proteins, and drugs. Biopharmaceuticals are therapeutic products produced by living cells rather than chemical medicines. Example: insulin, interferon, growth hormone, vaccines. Recombinant DNA or DNA technology allows scientists to modify the genetic material of organisms to produce these complex biological molecules in large quantities. Definition: recombinant DNA technology is a process of joining DNA molecules from the different sources and inserting them into a host to produce a desired protein. The host acts as a biological factory to synthesize the required biopharmaceuticals. Step 1. They involves some steps. Identification and isolation of gene. The specific gene coding for the desired therapeutic proteins, example insulin, is identified from human or animal DNA. Restriction enzymes or cDNA techniques are used to isolate the gene. The isolated gene becomes the blueprint for producing the recombinant proteins. Techniques used: gene libraries, polymerase chain reaction, PCR, restriction digestion, and gel electrophoresis. Step 2. Insertion of gene into a suitable vector. The isolated gene is inserted into a cloning vector to carry a DNA molecule like plasmid or bacteriophage. Vectors often contain origin of replication, ensures replication inside host, selectable markers, example antibiotic resistance gene, multiple cloning sites, MCS, site where the gene is inserted. The same restriction enzyme is used to cut both the vector and the gene so they can join by DNA ligase. Step 3. Transformation into host cell. The recombinant DNA is introduced into a suitable host cell such as E. coli, yeast, or CH4 cells. Techniques like heat, shock, or electroporation are used for gene transfer. The host acts as biological factory for protein production. Step 4. Selection and screening of recombinant cells. After transformation, only few cells take up the recombinant DNA. These cells are selected using selectable markers, example antibiotic resistance gene using PCR, polymerase chain reaction following hybridization, blue-white screening. Step 5. Expression of the gene. The selected host cells use their machinery to transcribe and translate the inserted gene. This results in the production of the recombinant protein. Biopharmaceuticals, promoters, and codon optimization improve expression efficiency. Step 6. Downstream processing. The produced protein is extracted and purified from the cells or medium. Steps include cell lysis, filtration, and chromatography. Finally, the purified protein is defolded, tested, and formulated as a therapeutic product. Bacterial DNA. Step 7. Large scale production. Fermentation of cell culture. Recombinant cells are grown in a bioreactor under controlled conditions, pH, temperature, oxygen, nutrients. Example systems: bacterial fermentation for simple proteins, mammalian cell culture for complex glycosylated proteins. The protein is secreted into the medium and remain inside cells. Step 1. Step genes. Step 1. Insert genes for each of the two insulin polypeptide highly expressed gene. Step 2. Transfer E. coli with the recombinant expression vectors and select transgenic cells by their antibiotic resistance. Step 3. Purify the fusion proteins. Step 4. Chemically remove insulin polypeptide from the beta-galactosidase protein. Step 5. Combine the polypeptide to produce functional insulin. Next, applications. Formulation and clinical applications. Clinical applications of biopharmaceuticals. 1. Treatment of diabetes. Recombinant human insulin humulin is used to control blood sugar levels in diabetic patients. Produced using E. coli or Saccharomyces cerevisiae. Replaced animal-derived insulin from pigs or cows, reducing allergic reactions. Second one. Treatment of growth disorders. Human growth hormone HGH produced by recombinant DNA helps treat growth hormone deficiency in children's dwarfism. Also used for muscle wasting disease and Turner syndrome. Third application: treatment of anemia. Erythropoietin (EPO) stimulates red blood cell production used in patients with chronic kidney disease or cancer. Chemotherapy induced anemia produced in CH4 kinase hamster ovary cells. Fourth application: treatment of viral infections and cancers. Interferon alpha, interferon beta, interferon gamma enhance immune hepatitis B, hepatitis C, and certain cancers. Example, interferon alpha for chronic hepatitis C. Fifth application, treatment of hemophilia, recombinant protein factor 8 and factor 9 are used to treat hemophilia and A and B respectively. Prevents bleeding episodes in patients lacking natural proteins. Cancer therapy, monoclonal antibodies made using recombinant DNA target specific cancer cells. Example, transformer accepting breast cancer, HCR2 receptor, human epidermal growth factor 2 receptors, rituximab, non-Hodgkin's lymphoma. Examples of biopharmaceuticals produced by RNA technology, recombinant DNA technology, biopharmaceuticals, gene source, post-used therapeutic use, human insulin, gene source, human pancreatic beta cells, post-used E. coli, Saccharomyces cerevisiae, therapeutic use, treatment of diabetes mellitus, human growth hormone, gene source, human pituitary, post-used E. coli, treatment, therapeutic use, treatment of dwarfism, interferon, genes and leukocytes, post-used CSO cells, teenage monster ovary, treatment used in viral infection and cancer treatments, erythroprotein, gene source, human kidney, CSO cells, therapeutic use, treatment of anemia. Advantages: high purity and safety, large scale and cost effectivity production, avoids animal and human contamination, possibility to modify